So, good evening, everyone. So, today is the week 10 of the live session of mostly focusing on uh, omics technologies, which is what the mass spectrometry based proteomics and NGS based technologies for genomics technologies. So, we would be starting with of which we had discussed in the week one in a uh, little bit manner regarding the shotgun proteomics. So in this week, we have seen that how for interactomics, if we try to understand or if we perform experiments like IPMS, that is immunoprecipitation, with followed by uh, so once you have a target molecule that you try to pull it down in your sample through antibodies, and then followed by if you perform the mass spectrometry. So the concept in the interactomics for understanding the interaction. So after you have pulled down and have done perform through the different bits, might be auto antibodies or might be some relevant antibody relevant antigens or something. So then you want to do the mass spectrometer. So understanding the concept of mass spectrometer again becomes crucial to interpret or to perform this kind of experiments where you need to have a thorough concept of immunoprecipitation, and then followed by mass spectrometry helps us to understand that which proteins or which interactors are there, and what is your ideal thing, or which one do you like to take it forward, and which you like to analyze. So this is what we can say now. So the basic principle lies in to have the peptide separation followed by creation of ions, and then to separate the ions and the detector. So to have creation of ions, move it, and generation, uh, separation, then movement, and then finally to detect it in the detector. So the basic principle lies very straightforward, where you have starting from the peptide separation. So since we are talking about the protein interactors, so it would be the protein separated, and then followed by which is creating the ions, generating and moving it within it. So followed by that separation, and then finally detected by ions. So the basic principle or the major principle that stays within it, that it is measurement of the MYZs by the mass spectrometry. So this is what basically stays in the final uh, conclusion, is that to have measurement of the MYZs through the mass spectrometry, or the understanding the what is the mass to charge ratio after it has been ionized and the charges have been generated, to finally separate and then detect the ions. So this is what today we will be looking into it, to finally understand the interaction of different experiments. Finally, what goes into behind the technologies like this. So now moving on, so there are two approaches of uh, this thing which we can talk about. So I think the slides are not moving somehow, so I will just reshare it once again. Right, so what we are getting, so top down versus bottom up approach. So what is the main difference? So there are two types of differences that can be seen. One is the top down proteomics. So we have the protein separation, proteoform secreted, and then finally in the MS2 level to find out the proteoform analysis. So this is requirement for the mass measurement of an intact protein. So if you have an intact protein and you want to perform it without breaking it down, So this is molecular mass database, search for identification of protein, and MSMS fragmentation of an intact ion, and this is related to the sequence specific ion search. Sequence specific ion search. Sequence specific ion search. So this is basically what the major difference between here and the bottom of is that. So here if you have an intact protein and you are injecting it into mass spectrometer to get the intact mass fingerprint of that particular protein, and then you are taking it for the identification of the proteins, and then that is followed by the MSMS fragmentation of an intact ion, and then to finally sequence specific ion search. But in the case of the bottom of proteomics, what we do or what we try to do is that we have the proteins which is followed by the peptides, and then we inject in the mass spectrometer followed by separation through a liquid chromatography the peptides, and those peptides are finally analyzed, and the peptides are then mapped back into the protein. So that is what actually lies the concept behind the bottom of proteomics, where the peptides are taken forward and mapped into the proteins. So for this, you require the triptych digestion, peptide mass sprints, and the database assured identification of the peptides and the proteins. So for here, so the major difference I think you have understood. One is that the whole protein is what taken forward and then analyzed in mass spectrometer, where the mass is measured of an intact protein, and the molecular mass database is searched for the identification of the proteins, and it is the MSMS fragmentation for that intact ion, and it is giving you the sequence specific ion search. Whereas in the bottom of proteomics, what is there is that the proteins are digested back into the peptides, and then peptides is separated, and once the peptides is separated and selected, then followed by that peptide analysis is performed, which gives you to, which is mapped back with the proteins, and to finally understand that what proteins are there. So that is the major thing of what we say as a triptych digestion followed by this. So for this digestion, there are 2-3 steps that are required. So if you want to do once you have done the in-gel or in-salt with the IPMS experiment and the interactions, so now what you are trying to do is that you are trying to do the bottom of proteomics by looking at the peptides, by digesting into the peptides, and then finally mapping it back with the proteins. So for that, what is required, or what are the things that are required is that one, just a minute. Yeah. So what it is required? So certain steps are required for proteins to digest, right? So first you will be extracting of the, yeah. So first I am just writing that you would be extracting the protein from sample. So that extraction depends on different sample types through different buffers. So extraction of proteins from the samples. So once it is done, what you will do is that you will do a quantification, right? So quantification you can do using various methods like Bradford. So you are doing a protein quantification. So you are quantifying the amount of the protein there, right? I think again there is a problem. The slides is not moving. I'll just reshare it once more. right? So the quantification of the protein. So now you will be taking a specific amount of proteins for digestion, right? So you will take a specific amount of protein for digestion. That at how many micrograms of protein you will be taking for digestion. So these proteins are, you know that these are folded, and for that you need to treat them with CEP or DTT, which are reducing agent. So what it does is that, the disulfide one gets broken down. So we do one thing, insert, right? So let me explain the process once very clearly. So these are protein which you are extracting. Then you are doing what? You are adding trypsin. Sorry. So you are quantifying. So you are taking that protein, certain UG. I'm writing certain UG. And then what you are doing is that you are quantifying. So you are treating it with TCP. So what TCP does? So it reduces disulfide linkage okay. So it reduces the disulfide linkage SS bond to SHSH bond. So once it is done, so it is then treated with IA, iodoacetamide. So what it does that it make it to CH3, that is your alkylation step. So this alkylation, what it does is that it does not let the reversible reaction of disulfide linkage, that is the reduced state, to again be formed back. So reversible reaction of uh, going back to disulfide linkage is not there, right? Once that is not there, that is alkylation is not there, then so why this is seen? Because this is the protein structure. If it is in a folded state with the disulfide linkages and all, then the trypsin cannot go and bind and go and act on it very easily and very firmly. So what reversible reaction by going back and forth is reduced, and when it has been broken down, it has been the sulfur certificate. Then trypsin is something that is added, which cleaves after arginine or proline. Sorry, after arginine or lysine, followed. So it again depends on the trypsin. If trypsin is of not of very high standard quality, then what happens is that after arginine or lysine, if there is a protein, then there is a bulk effect, or that some uh, hindrance from this bulky nature of the protein group, which does not let the trypsin to cut it very efficiently, and that's why uh, what it uh, and what it does is that the trypsin does not uh, get it cleaved there. So once it is done, then finally you have the peptides ready for taking it forward in the mass spectrometer. After, definitely the peptides needs to be desalted and cleaned. Desalted and cleaned up. Right. So peptides needs to be desalted and uh, uh, okay. So once the peptides are cleaned up, then what we can do is that uh, desalting and the clean up stage is done, then we can, it is ready to put it into the mass spectrometer, right? So this is the overall concept behind the major thing behind the bottom of proteomics for to digestion and to inject it in the mass spectrometer, and finally the peptides are mapped backwards towards the protein for finally uh, getting the result, right? So this is what finally you can tell of or consider of uh, for the analysis, right? So any question or any query till now? No, right? So this is what we look for in the bottom of proteomics. We have been taking the samples, quantifying the proteins, or digesting it through. Before going to that, we are treating with the CP and IA for reducing it, and then finally alkylating it so that the reduced disulfide linkage or bond does not get back attached to it, and then finally cleaving it uh, through the peptides for you to take it forward there, so that peptides can be taken forward for injecting into the mass spectrometer. But the major problem or major thing that lies is that you need to clean up your peptides, and you need to understand, but how? how you are understanding, or how you are understanding that whether the this is a particular peptide and how the algorithm identifies it. I think again the slide is not moving for some particular reason. Let me just reshare it once more. Right? Yeah. So why we are talking about tandem mass spectrometry? So we have understood some of steps of the sample preparation and why it is required and why it is done. So specifically, this is catering towards our need for understanding the experiments like immunoprecipitation IPMS or immunoprecipitation based mass spectrometry. So now what it is doing is that, so just understand the concept. So just is that it's MAC, which we can understand, right? So MS, the sample is there. We are ionizing EISI multi. So different ionization techniques, which we will talk to some extent, and we have discussed it in week 1 2 with a greater extent of the different hard ionization techniques and the soft ionization techniques. How the scientists have decided and when we could control the ionization. That was revolutionary in the field because we could use it in for various studies and for various applications, right? So the ESI multi, which we are in, then we are doing the M/Z separation, which followed by the fragment, and then the MS2 or the product ion. So this here there is one fragmentation, followed by here another fragmentation, and then finally it is detected. So why it is two? So that's why it is known as tandem mass spectrometry, or uh, tandem mass spectrometry, or two types of mass analyzers are required for uh, analyzing the peptides. One is at the MS1 level and one is at MS2 level. So the major thing is that, so the MS1, it is helps in identifying the molecular weight of a biomolecule. So if you have the peptide, you have the precursor, it will be helping you to identify the MS1 for the molecular weight of the biomolecule, whereas followed by MS2 is reduced required for reducing the peptide sequence. So I'll just explain it why and how in just a minute. Sorry, there was some technical issue. Yeah, so why we require uh, these two amino acid, right? So if we are giving one peptide, right, this is a full peptide with CONH bonds. So once we get it, so at this level we will get the M by C of this peptide, right? But what is the sequence of the peptide? We don't know. And if we don't know the sequence of the peptide, then what will happen is that we cannot go and try to understand and map with the proteins. So for that, the two mass analyzers are required, and this hybrid configuration are required to identify the peptides. And the Y ions are generated from the C terminal, and the B ions are generated from the N terminals, which are mostly frequent and visualized and gets identified there. Whereas the X, so these are the ions which is X, Y, Z and ABC ions are generated, but this B ions and this Y ions are mostly frequently generated, which are identified through Y1 minus Y2 and B7 minus B6 are taken forward. So here, if we look at the uh, overall workflow, so this sorry. So if we look at the overall workflow of the things, so I think the slides are not moving. So I'll just do what is that unshare and reshare it once again. So yeah, so which we have discussed, right? So tissue. So here the steps like PE or protein extraction. Reduction of population fixing. Right. So here, so what is happening is that we are requiring the mass spectrometry based. So these peptides, which we are. So this is just an overview of what we have discussed till now. So we can see that this peptides and is the reverse phase liquid chromatography, then which was by the LC-MS, and then finally that is the MS spectra. So these are the ions of the N-terminus B type ions and the Y type the C terminus ions. So this is what is an overview of what we are talking about when we are trying to talk about IPMS experiment with the MS component being mostly the shotgun LS2 other than the mass spectrometry. So you can go through this paper of the analysis of proteins and proteomes by mass spectrometry. So if you want, I can share this thing. Right, this one. So what is written that the decade after the discovery of electrospray and matrix-assisted laser disruption ionization methods that finally allowed gentle ionization. So again, we had discussed that game changer was the gentle ionization of the biomolecules, and the mass spectrometry has become a powerful tool in protein analysis and the key technology in emerging field of proteomics. Right? So the success of mass spectrometry is driven both by innovative instrumentation design, especially those operating on the time of flight or ion trapping principles, and by large-scale biochemical strategies, which is mass spectrometry detect the isolated proteins. Any human protein can now be identified directly from genome services on the basis of minimal data derived by mass spectrometry. This has already happened in genomics. Increased automation of sample handling, analysis, and interpretation of results will generate an avalanche of qualitative and quantitative proteomic data. Protein-protein interactions can be analyzed directly. So here is the importance of what we are studying in the course of Interactomics, that the protein-protein interactions can be analyzed directly by precipitation of a tagged bait followed by mass spectrometric identification of its binding partners. So the bait and the prey concept of which of identification can be done. By these and similar strategies, entire protein complexes, signaling pathways, and whole organelles are being characterized. PTMs remains difficult to analyze, but are starting to into it generate strategies. Then we have the mass spectrometry is a venerable technique whose beginnings date back to the early days of the last century. Among the analytical techniques, MS holds a special place because it measures the intrinsic property of a molecule, its mass, with very high sensitivity, and therefore it is used in an amazingly wide range of applications. Beginning on the 1880s and a larger scale in the 1990s, mass spectrometry plays an increasingly significant role in the biological sciences. Uh, why is it taken so long? So mainly because mass spectrometry requires charged gaseous molecules for analysis, biomolecules being large and polar, however, they are not easily transparent to the gas phase and ionized. So the problem was that the biomolecules are big, large, and they have very much polar, easily transferred. Electrospray ionization and matrix laser are the ionization techniques for the success. This method was developed and were the basis for increasingly power that became available a few years later. Major advances were so made in the sample preparation for MS, a crucial area for overall feasibility and sensitivity of analysis. Starting in 1993, software algorithms were published. Mass spectrometric data obtained for protein with increasing populated sequence databases. In retrospect, this event marked the transformation of mass spectrometry to a large-scale functional genomics technique. The last few years have seen development of even more powerful instrumentation and algorithms for protein characterization, a trend that shows no signs of slowing down. At the same time, MS was being developed to meet the demands of molecular biology for high sensitivity. The concept of proteomics began to be popularized. Proteomics is defined as a large-scale analysis of the function of genes and is becoming a central field in your functional genomics. A major tool to study purified in this field is mass spectrometry. The history of proteomics dates back to the discovery of two-dimensional gels in the 1970s. First feasible way of displaying hundreds of thousands of proteins on a single gel. Identification of the spot separator remained laborious and was limited to the most abundant proteins until the 1990s, when biological mass spectrometry had developed into a sufficiently sensitive and robust technique. Today, mass spectrometry is an essential element of the proteomics field. Researchers are now successfully harnessing the power of MS to supersede the two-dimensional gels that originally gave proteomics its impetus. Currently, the use of MS in three major areas. MS is the preferred technique for characterization and quality control of recombinant proteins and other macromolecules, an important task in the field of biotechnology. It is also commonly used for protein identification, either in classical biochemical projects or in large-scale proteomic ones. Finally, because MS measures the molecular weight of a protein, it is the method of choice for the detection and characterization of post-translational modifications and potentially can identify any covalent modification that alters the mass of a protein. So, describe the principles of the MS ionization methods, major types of instruments, and under development strategies for the intact proteins, and what the sequencing of peptides obtained after enzymatic degradation of protein, and cover the identification of proteins in databases by MS data. Analysis of PTMs and the recent development of quantitative mass spectrometry using high stable isotopes are also discussed. Of equal importance to the technological developments is the development of strategies for applying MS to solve real and interesting biological problems. We discussed the difference between expression proteomics, measured upregulation, downregulation of protein levels, functional proteomics as exemplified in several recent strategies aimed at the characterization of cellular compartments, multi-protein complexes, and signaling pathways. So, ionization methods, if we look at MALDI or matrix assisted, I think again it is not moving. I'll just share it once again. So, matrix assisted laser disruption ionization or MALDI, developed by Karas and Hillenkamp in the late 1980s. One of the two soft ionization methods to generate your gas phase protonated molecules. A large excess of matrix material is co-precipitated with analyte molecules. There are some molecules to be analyzed by pipetting a sub-microliter volume of the mixture to a metal substance and allowing it to dry. Solid is then irradiated by nanosecond laser pulses, small nitrogen lasers with a wavelength of 337 nanometer. The matrix is typically a small organic molecule with absorbance at the wavelength of the laser employed, worked with biomolecules almost exclusively. This is matrix of alpha cyano-4-hydroxycinamic acid or dihydro benzoic acid DHB or for alpha cyano-4-hydroxycinamic acid. Differ in the amount of energy they impart during the disruption and ionization, and hence the degree of fragmentation. The alpha cyanomatrix, which generally leads to the highest sensitivity in MALDI, is hotter than DHB or dihydroxybenzoic acid. So latter is preferred when the ions need for the stable of milliseconds in trapping experiments rather than microseconds in time of flight experiments. Numerous variations on the basic fried droplet method of sample preparation from MALDI have been described. This can be laid down in a microcrystalline thin film on the substrate with to better adherence, so the sample can be washed more vigorously. A larger crystalline surface from which ions can be dissolved. Administered with the matrix can be beneficial. Nitrocellulose includes peptide and fructose can lower the energy state of the dissolved ions. The precise nature of the ionization is strictly largely unknown. Signal intensity depends on incorporation of the peptides into crystal, the likelihood of capturing and retaining a proton during the dissociation process, and a number of other factors. Other peptides with the C-terminal arginine generally result in higher signal than peptides with C-terminal lysine. For this reason, it is difficult to relate peptide peak height with the quantity of sample. An internal standard used. 500 aldons is often obscured by matrix-related ions to some extent, and broad peaks and losing with multis to separate the analysis of peptides. Yes, electrospray mass spectrometry developed for the biological by Fain et al. Liquid containing the analyte is pumped at low microliter per minute flow rates through a hypodermic needle at high voltage to electrostatically disperse or electrospray. Electrostatistically disperse or electrospray small micrometer sized droplets which rapidly evaporate and which impart their charge onto the analyte molecule. This ionization process takes place in the atmosphere and is therefore very gentle without fragmentation of analyte ions in the gas phase. The molecules are transferred into the mass spectrometer with high efficiency for analysis to stabilize the prey and nebulizer gas, and some other devices often employed. A wide range of compounds can be analyzed by ESMS. The only requirement is that the molecule is sufficiently polar to allow attachment of a charge. So this includes proteins, oligonucleotides, sugars with this sensitivity as sodium rather than hydrogen, and polar lipids. For a given compound, the signal strength peak height in the spectrum increases linearly with the analyte concentration over a wide range until saturation occurs. However, the signal is to a first approximation independent of liquid flow rate, which makes it desirable to operate at the lowest rate possible. I think there is again some issue. So there does not seem to be an upper mass limit to analysis by ESMS. Large ions are typically multiply charged. Proteins and peptides by added protons in the positive mode and abstracted protons in the negative mode, which brings them into the range of mass to charge ratios of typical mass spectrometers. This is other than a mass. Okay. So range of mass to typical mass spectrum. The distribution of charges gives rise to the typical multiple charge envelope. This spectrum can be simplified by. So what happens is typically multiply charge put in the peptide where in the positive mode and etc., which brings them into the range of typical mass spectrometers. So m by z rather than a mass. So if they are highly charged, it brings into a range. Distribution of charges gives us a typical multiple charge envelope. This spectra can be simplified by deconvolution and algorithm that sums up the signal intensity of signal peak at the molecular weight of the analyte. Very complex mixtures, increasingly difficult to interpret the components of the numbers. This is typically performed with either the infusion mode, the electric spray format, or combination with the high performance liquid chromatography. Right? So it is either performed in the infusion mode in the infusion mode, sample is simply introduced into a continuous liquid stream, typically a mixture of organics such as 50-50 methanol by an injection valve. Valve flow rates will vary between 0.5 and several microliters per minute. Several have to be substantially free of salt and detergent, but can conveniently be cleaned in a reverse-phase packing loop in the injector valve. Nanospray is a miniaturized version of electrospray that operates without pumps and at very flow rates in the range of few nanoliters per minute. It is without the pumps. So it is performed in pooled gas capillaries. Pooled gas glass capillaries with an inner diameter at the tip of about 1 micrometer. A microliter volume of sample can be analyzed for more than an hour at full signal strength, which allows complex sequencing experiments to be performed. Okay, so it is performed in pooled gas glass capillaries with an inner diameter the tip of about 1 micrometer. Well, liquid chromatography and mass spectrometry are coupled. Mass image analysis of components of the sample takes place on one line as they look from the chromatography. In this scenario, sample and separation considered are achieved in a single stage. The last year has been tremendous in mass spectrometry instrumentation. So it is interesting to note that the cumulative advances in MS instrumentation have occurred at a much faster pace than the increases in computer processing speeds. This progress is the result both of incremental advances in existing mass spectrometers and of entirely novel combinations of existing instruments. Essentially, all mass spectrometers measure the mass to charge ratio. Protein, peptides, or proteins. Three different principles may be applied to achieve uh, on the basis of time of flight or separation by quadrupole electric field, so which we will be discussing in some time. generated by metal rods, that is quadrupole MS, or separation by selective ejection of ions from a three-dimensional trapping field, ion trap. Separation by selective ejection of ions from a trapping ion trap or Fourier transform ion cyclotron MS. For structural analysis such as peptide sequencing, two steps of mass spectrometry are performed in tandem mass spectrometry or MSMS. So for structural analysis, which can be done by employing the same separation principle twice or by combining two different MS separation principles. Both multi-ion can be coupled with any of these three methods of separation. The multi produces a short burst of ions in the vacuum and continuous beam of ions, typically lead to coupling with multi with TOF, quadruple and ion trapping MS. Multi TOF. Schematic combining multi and TOF in a mass spectrometer, samples are deposited to metal substrate, folding into separate. So, multi and tau samples are deposited, spots are then irradiated by laser pulse in short bursts of ions. The ions are accelerated to a fixed amount of kinetic energy and travel down a flight tube. These small ions have a higher velocity and are recorded on a detector before the larger ones, producing the time of flight spectrum. Several dozen to hundreds of laser shots are averaged. Dozens to hundreds of laser shots are averaged to produce the final multi-spectrum. Performance in modern reflector is typically the range of a few parts per million in mass accuracy and only about a femtomole of peptide metal is to be deposited multi-directly to produce a signal. Decay because of the energy departed in the desorption process is to obtain structural information and technique termed post-circuit can provide important structural information but does not serve as a general peptide sequencing method which is unsubstantially sensitive and simple to control. Then electrospray quadrupole mass spectrometers is most often combined with a quadrupole mass spectrometer. This quadrupole is a mass filter, four rods to which an oscillating. So this is the multi-TOF mass analyzers applied unless only certain mass pass through the other mass on unstable trajectories and do not reach the detector. By scanning the amplitude of the electric field and recording the ions at the one obtains the mass spectrum by scanning the amplitude of the electric field and so what we are that and which is only a certain mass pass through the other masses on unstable trajectories. So is the detector by scanning the amplitude of the electric field and recording the ions at the detector, one obtains a mass spectrum. Most peptide sequencing have been performed that consist on masses separating quadruples separated by central quadruple or had multiple sectors, so it obtain the ions yielding fragmentation. So quadruple are capable of unit mass resolution and mass accuracy of 0.12 or 1 Dalton and excel at quantitative measurements owing to the presence of two independent can be programmed for scan modes in addition to the isolation of a peptide followed by obtaining a mass spectrum of the fragments. Right? Passing rings Q0, Q2, Q1, quadruple top instrument, ions are produced at the atmosphere and Q top the ions enter the atom system. Like this can be said Q1 and dissociated in Q2. In the time of flight, both read and pulse on the reflector onto the detector. Triple quadruple has begun to be complemented by the Q top for quadruple time of flight, in which the third quadruple section is replaced by a top analyzer. Its main advantage is over the high accuracy on mass and high resolution typical of modern time of flight instruments, unambiguous determination of charge state, and very high specificity in database searches. So now is the ion trapping instruments. Ions can also be trapped for analysis in 3D electric field. This ion trap captures of ions up to the limit of their space charge. This is the maximum number of ions that can be introduced into the instruments without distorting the applied field. The ions are then subjected to additional electric fields which eject one ion species after another from the trap and are detected to produce a mass spectrum. For MSMS, all except the single desired ion species are ejected first and species fragmented and processed, and several sequential steps of MSMS can in principle be performed along digital fragmentation studies owing to the operating principle of the trap, the lower end of the fragment mass spectrum. Impact and versatile whose operations are slightly automated. Another version of the trapping principle is embodied in the MS teams that the ions are captured in a combination of electric field and a very strong magnetic field. Looking at standing complex instrument is not currently widespread use in biological mass spectrometry. So other mass spectrometers, the main types of mass spectrometers, for example, multi-quadruple TOF instrument consists of multi-ion source coupled to the quadruple TOF analysis. The attraction of this instrument sufficiently sensitivity for demanding proteomic applications, ability of multi to analyze a large number of samples with the ability to sequence and receptive peptides, or multi produces far fewer ions than electrospray, and then they are singly charged, which leads to fragmentation patterns that are more complicated than those of the typically multiply charged species. Multi can also be combined with a two-section TOF instrument separated by fragmentation chamber, so-called top arrangement. This instrument should have the advantage of a very fast transfer in the case of the multi-quadruple top, low number of multi-produced ions, additionally more difficult to obtain high resolution, highly accurate segmentation spectra after an initial top separation compared to this well-defined ion beam produced by the multi-quadruple top instrument. So now is the protein purification and the preparation. So the upfront isolation procedures can have the most significant impact on the outcome of an MS-based investigation. For example, sensitivity of the overall procedure is usually determined more by purification strategy than by the sensitivity of the MS instrument per se. Typically, protein purification starts with a wholesale lysate and ends with a gel-separated protein by the spot. MS analysis is usually carried out on peptides obtained after enzymatic degradation of these gel-separated proteins. So the intact proteins are analyzed or the gel electrophoresis step is omitted. And it is a special consideration for protein separation methods be any of the classical separation methods such as centrifugation, column chromatography, and affinity-based proteases can precede the final gel electrophoresis. Generally, silver stained amounts are necessary for the successful identification of proteins. But even higher sensitivities have been achieved by a specialized rule. It is important to minimize contamination with keratins, which are introduced by dust, chemicals, handling the products easily dominate the spectrum, and salts are incompatible with 2D gels and mass. If a dialysis of the sample may be required, if the protein can be eluted from the best sample preparation is achieved on small low pressure traps that can be incorporated in injection forms. In affinity based protocols, this is such as compared, for example, bacterial proteins in a protease fusion preparation will hinder analysis. So again, in the relevant from the interactomics point of view, that it should be done. Now the traditional preparation for gel separated proteins. Most of the detergents are eliminated. Nevertheless, the protein should be now concatenated, right? Uh, Concentrated as possible in the gel to avoid excessive background in the MS analysis. So pooling of spots is not necessarily advantageous, as both protein and background will uh, increase. Chromatic staining, silver staining, or radioactive labeling, which are all compatible with MS analysis, can visualize proteins. Cross linkers and harsh oxidation should be avoided as they interfere with the extraction of peptides. Chemically modify the peptides. Silver staining provides adequate sensitivity, which should be components with a narrow linear range. Protein bands are excised from the gel and subjected to reduction and condension of several processes, and finally inject protein deletion followed by peptide extraction. A small portion of the resulting can be directly used for multi-peptide mapping. After for multi-peptides are desalted concentrated, this can be performed by columns of diverse material in nanospray needles or gel floatatives by injection into the liquid chromatography MS columns. Due to the multi-targeted nanospray alluded to microcapillary convergence interacts several the mass spectrum recombinant proteins, glycosylation, full bending of therapeutic growth factors can be studied. Precise molecular weight of the major and minor forms of the protein data that cannot always be gained from peptide mapping. However, large proteins are typically heterogeneous, making molecular weight determination difficult. Electrospray of choice of determining of proteins as multi results in broad peaks and low sensitivity for proteins above 30 kilodalton. I'm sure positivity of detergents usually accomplished by reverse test chromatography. For me, that it can be used. So I think again there's an issue with the screens. So yeah, so the formic acid can be used as a solubilized protein for electrophoresis. So the mass spectrum of small protein are not high use solution instrument. Identification of intact proteins with cellular sizes with molecular weight analysis is difficult for several reasons. Sensitivity of ESMS for large molecules is poorer than for peptide analysis because the signal is distributed over many charged states and the heterogeneity of the protein similar splits the signal into many components. More fundamentally, the molecular weight of a protein cannot be predicted precisely from its database entry, NFC terminal processing, PTMs, and chemical modifications using sample purification. Therefore, even a precise molecular weight version not identification of the nevertheless molecular weight version not allowed into nevertheless in a recent study several hundred recorded on a single experiment. Of the above mentioned results. Peptide sequencing by the tandem mass spectrometry of what we were talking about. The sequence of peptides can be determined by interpreting the data resulting from fragment tandem mass spectrometer. One peptide species out of a mixture is selected in the first spectrometer and is dissociated by collision with an inert gas such as argon or nitrogen. The resulting fragment is separated in the second part of the tandem mass spectrometer producing the tandem mass spectrum or MSMS spectrum. The instruments are used to do multiple collisions impart energy into the molecules. This is how low energy fragmentation with any single hit is not sufficient to break. And single hit can be several bonds along the backbone can be broken. The most common at times are the P and the Y, which fragmentation of the white bond with change retention A not C terminus respectively. Most peptide sequencing is performed on electrospray dyes, have a general state corresponding to the number of positively charged amino acids plus the charge formally located at the N-terminus of the peptide. Often doubly charged or triply charged if they contain a histidine residue. Larger peptides are multiply charged. Fragmentation often contain multiply charged answers with computer assistance or matched against databases in high variability spectra. It's possible fragmentation ladders from the low mass and through the highest mass ion. For example, the YN series of triptych vector with masses 175. The next fragmentation YN series referred by mass of an amino acid residue was spells out. And next amino. Similarly, BN starts with a B1 and is traced upward in molecular weight. A priori, usually a complete set of VNs will doubly confirm the entire two-pair sequence. Not all fragment ions are present. Okay. Okay, not all fragment ions are present at the detectable levels. Arise by double fragmentation to well goes. That is often possible to interpret part but not all the sequence with confidence. Other features of the MSMS spectrum include ammonium ions, double cleavage of the peptide backbone, intermineral centriminal. These ammonium ions can indicate or confirm the presence of individual. And fragmentation is not equally likely along the entire length. For example, fragmentation between the two N-terminal amino acids is energetically unfavorable. The P1 ion option is not involved. Two N-terminal amino acids is energetically unfavorable. Okay, and therefore the B1 ion is often not observed. So why B1 ion is often not observed? B2 ion, however, is companion. The A2 ion it at 28 Dalton. CO mass difference is usually very prominent. A2 ion and the B2 ion, but energetically unfavorable in the B1 ions. Likewise, C-terminal bond of a proline residue is weak, but cleavage at the N-terminal residue is very prominent. Another feature that frequent occurs is switch over from a C-terminal to N-terminal series because of charge retention of positively charged such as histidine. So the bond breakage of the doubly charged peptides often is important characteristic to proceed by the localized charge on the arginines or on the C-terminus and the delocalized proton located on the bond breakage along the backbone, the arginine. So bond breakage of the doubly charged triptych peptides. The doubly charged triptych peptides, most often used in protein cutters, believed to proceed by the localized charge on the arginines or lysine residue on the C terminus and the delocalized proton formerly located on the N terminus, which induces the strongly localized proton peptides containing internal arginines, often resulting in a therapeutic development. Internal arginines are very therapeutic. As mentioned, a fragmented large multiply charged ions of a list multiply charged ion series. Advantages in studying this answer is direct assignment of the charge of the spacing of the carbon isotope. They space one third of a dalton. Their triply charged larger open fragment. High resolution is of advantage in studying this answer because it allows direct assignment of the charge state of fragments based on the spacing of the carbon isotopes. Distinction between isoleucine and leucine, which has the same mass, so the distinction can be principally by using the different retention time of the containing peptides during chromatography or by the side chain fragmentation in high energy collisions. So, high resolution studying this as is allows direct assignment of the charge state of fragments based on the spacing. There's a triply charged species, so Multiple choice is a high resolution, multiple charge time series, and the high resolution advantages for which allows direct assignment. So, larger peptides often fragment efficiently and provide long ion series. It's distributed over several constituent obvious high precursor which is over several charge states. So, just give me a minute. So, just a minute, I will start again. Yeah, so I think there's again issue with the technical issues which we are facing now. I'll just protein identification by database searching. A key advance in biological mass spectrometry was the development of algorithms for the identification of proteins by mass spectrometric data. Master database using a set of peptide masses and now increasingly using fragmentation spectra of the individual peptides. For this reason, described in previous sections. Uh, but this is representing the complete set of sequence of peptide was time consuming with the availability of the complete sequence of the model species and peptide sequencing problem. As we now in automation, the scaling of proteomics system is. Method of mass fingerprint is obtained enzymatically decreased, such as trypsin, typically obtained when compared to theoretically expected 50 peptide masses. The proteins can be ranked according to the number of peptide masses, mass accuracy, and the percentage of the protein sequence covered, and attempt to calculate the level of confidence for the match. Other factors can also be included, such as fact, uh, less frequent The accuracy obtained in the measurement of peptide mass often influences the specificity of research. High mass accuracy is achieved at the rule at least 5 peptide masses into match to the protein. 15% of them needs to be covered for an unambiguous application. After a match has been covered, second mass search is performed. Also resolve simple protein mixtures consisting of match detector, remove the peptides. Accumulatively peptide mass fingerprinting is used for representing a single protein component. So now searching with the tandem mass spectrometric data. So databases can also be tandem peptides. Can be combined with the start mass and which is the procedure can be automated. Other methods to access tiny sequence information. The experimental spectrum is matched against full of peptides in the database. Score is given to determine. The other score indicates how differently the next similar sequence. Although this method can be highly automated, sequence is to be verified. In the sense of an ANDRA, or 10 to 20% estimator can be required to further interpretation. Large percentage can be verified manually. Tandem allows different analysis of protein mixtures, one-dimensional yields. Software reports a list of proteins, one or several peptides, group protein mixtures, contiguous peptides, and searched in databases. Searched by mass spectrometry data, non-redundant protein. So this is fine. Database searching for modifications. Data attributes or their few will be modified. There are only a small number of peptides are required. Modifiers also discovered. Peptide sequence in the database is additional to a phospho group. CMS is a powerful technique. This method will read different separations and sensitive identification of the individual components of mass spectrometry. Hundreds of proteins can be analyzed directly even when concentration levels of different proteins may also sensitive to combination with 1D or 2D immunoprecipitation or other protein purification techniques. Again, comes into handy of our thing of immunoprecipitation. Although numerous methods for coupling LC to mass spectrometry have been explored, it is electrospray ionization that has transformed into routine laboratory process essentially unit to analyze protein levels. Interesting biological research. ES requires a continuous flow of liquid and signal strength is independent of the flow rate. As research efforts are focused on coupling nanoscale LC, a sub-microliter flow rate to highly sensitive microscale ES interface. Nanoliter per minute flow rate, also called micro ES, micropicillary LC or picospray. Currently, detection limits of few femtomoles of peptide material loaded onto the column make this technique compatible with silver cell fluorescent massive gel bands and capable of per cell. Eluted from a reverse phase column to separate the peptide research and is ionized and transferred with high precision mass spectrometer. An individual species in a complicated generator, for example, the peptide ligands associated. For example, the peptide ligands associated with the human measured histocompatible complex 1 molecules are a mixture of across 10,000 different peptide species. Representing the amount isolated was loaded into nanometer and trap mass spectrometers and longer mass spectra were acquired. There are 2000 the acquired contain molecular weight information on the peptide species and their amounts. Scan was summed and plotted as a function of chromatograph. The data acquired contain molecular weight of the vectors in the ion current for each scan. Ion current can be summed and plotted as a function of time. This is to the total ion current chromatogram. Post-acquisition data can be interrogated to reveal the ion current recorded at a particular MBZ. Post-acquisition, the data can be reveal the ion current recorded at a particular MBZ. or selected ion chromatogram. Current chromatogram. The ion current recorded at a particular M by C molecular weight or a selected ion uh, can be sequenced without further purification by inserting the routine peptide, fragmenting it, and obtaining a spectrum. In this manner, large number of peptides can be sequenced in single sequence with that of the same molecular weight if they differ in hydrophobicity. In practice, the mass spectrometer is often programmed to perform one scan if they differ in hydrophobicity. Mass spectrometer is often programmed to perform one scan to determine the peptide mass and then to sequence the 3 to 8 most abundant peptides. Acquisition separation principle is almost always reverse phase high performance liquid chromatography as applied elsewhere in protein chemistry. The only difference is that its dimension and the fluorescence are much smaller. With width of 30 seconds, typical peak width of 30 seconds. Higher separation efficiency is stored in principal batches with the coated tubular capillaries. A small amount of capillary zone is another technique not in general use. NanoLC once thought to be a specialized and difficult technique as it can routinely proceed in many laboratories over the low fluorescence separative by conventional transparent illumination is a three-column splitting device. Typical NanoLC columns are interdiode filled with the polymerase C18 coated stationary cases. As the column diameters of 75 micromolar are the current compromise between ultimate sensitivity and trouble-free operations. Columns are available from commercial vendors or are packed in individual laboratories, and then conventional sample can also introduce column level that injects some micrometer to minimize losses while handling also an alternative displays the sample directly from the sample tube. Make between the liquid and power supply the charges. So recent improvements in instrument sensitivity, software control, automation, and data analysis tools provides unique high throughput capabilities for protein analysis when using LCMS. One approach is based on the proteolytic digestion of mixtures of proteins and LCMS analysis. Because peptides are now scrambled or multiplexed, it is no longer possible to identify proteins based on peptide mass proteins in tandem mass spectral data must be generated. Let the access of occurring SMS data, for example, in a typical data dependent acquisition, mass spectrometry full scan mass spectra to determine the molecular weights of the peptide species present in networks on 3 to 5 peptides. It takes less than 50 seconds to complete. In this manner, hundreds of ways can be generated. So it reduces the complexity of the peptide mixture, direct analysis of the yeast ribosome complex, offline and exchange chromatography and long gradient analysis, and in more than 500 deals in the East textile using high stringency for the database identification. Protein phosphorylation, so this we can do that later. The mass spectral signal cannot be easily to be the amount of analyte present in the sample. Our meaning proteins for knowledge of quantity changes is required. These changes are evident from the staining intensity of proteins and gels. Stable isotope method for small molecule mass spectrometer introduced image-based proteomics. Relative changes to the principle of these methods. Stable isotope derived and in one of the two states to be compressed. Stable isotope achieves the mass of the protein with predictable amount. The ratio of analyte between the two states can then be accurately. First application of the principle in microscope or either in normal peak ratios. Right. So moving on to our, so this was all about the proteins and the proteomics by the mass spectrometry. So now will be, so I think you have one through understanding for this. So now there are two kinds of quantification of what we were studying into it mostly. One is for the level free quantification and one is the level based quantification, of which level free quantification. So recent advancements in the improvement. So this, so again the slides are not moving, I guess. Just a minute. Yeah. So label free quantification, like if it is injected in the several time, so it can be through uh, higher intensity or through which the spectral counts that how much it's there, or if it is injected, the amount with the area under the curve, one can get the quantify it. And label based quantitation was that you need to use different labels or like the reagents, like I tried for this thing again. I think there's an issue with the teams. Yeah. So either you have the labels and then you are matching the reporter ions to the different levels. So this is the skeleton of the mass spectrometry: creation of the ions, separation of the ions, and the detection of ions. So ionization procedures are like in electrically charged molecules. Ions are accelerated by an electrical field. Their speed depends on the ion's charge and the weight. By measuring the time it takes the ions to pass a certain distance, uh, and the incidence of different molecules in a test can be determined. It was impossible on a large molecule such as protein before large ions could be produced in gaseous forms. Koichiro Tanaka showed that the laser pulses could blast protein molecules from a produce. So hard ionization techniques, higher energy resulting in greater amount of fragments and low number of molecular ion, low energy resulting in low fragment ions, and high molecular ion. So that is ESI or multi. So the measurement of the mass is by mass spectrometry, right? So what we can, that m plus h is mass plus 1 divided by 1. M plus 2h is something m mass plus 2 divided by 2. M plus 3h is mass plus 3 divided by 3. M is equal to ion mass and the Z equals to charge of ions. And Maldives matrix assisted laser disruption ionization that produces single charge ion species resulting in large embassies ion species. So we have gone through a lot of the different mass analysis at the time of flight, quadruple time of flight, but let us look into this mass analysis. So, but we are talking about time of flight mass analysis. So we have an ionization area. Now, if we look into detail of what we were studying of the different configurations of the mass spectrometer, then taking it forward for the analysis. So one is like the ionization area, then is acceleration area, then ion drift area, then the detection area. So this is a complete architecture of the time of flight. So quadrupole or time of flight there uh, included also. So ionization area, then followed by acceleration area, then ion drift area. So where the heavy ions and the light ions will fly on the basis of their respective MBICs, the light ions will be reaching to the reactor first, whereas the heavy ions will be just reactor second. So detection areas so separates ions and measures their MBICs based on the time they take pass to fly from the source to the detector. So this is the quadrupole or the hybrid configuration of the two types of muscle is very required than one what we had been talking. Eight. So this is a quadruple. So this is the two opposite roads with the plus potential and two roads with negative potential. Right. So what we are saying that two opposite roads with the plus potential and two roads with the minus potential, which acts as a filter only ions of a certain mass to charge ratio of passing of a certain filter. So what it happens is that it acts as a filter along all the ions of a certain mass to charge ratio pass for a. Okay, so the two quadruple. So this is where the two rods are there, acts as a filter, allowing only ions of a certain mass to charge ratio pass of a certain filter. And this is the orbicap and in an oscillating electric field, which is have not talked about there in detail in what the document we have shared. Rather, this is where it is happening in is happening in the oscillating electric field. And then finally is the ion traps. So similar, which we had also talked about, trapping the ions selectively based on the M by Z charges, and in the orbit trap you have the selectivity, and you have gone through it in the uh, lecture also, that how the is there and how the ions get separated. So then the concept comes of the resolutions, mass spectrometry resolution. I think we have gone through when we were looking at the document, which is the basics of to understand the mass spectrometry based proteomics. So that the resolution, isotopic peaks, and the charge state. So the M by Z difference between isotopic peaks is 0.5, and this is difference. So this is how the 2000 resolution. So for example, you can see that at the resolution, the two peaks can be two different masses can be uh, determined, and at what images it can be determined. So here is the formula of the understanding the mass resolution, that is the m by delta m, that is at the full width at half maximum, that the things are required for the resolution to change. So dimension is ratio of m by z value of a peak divided by its width at the half maximum intensity, so that to get at what the two differences can be done. So the mass accuracy, the exact mass, observed mass minus the theoretical mass divided by theoretical mass into 10 to the power 6. And these are the CID, or the collision-induced dissociation, of which it was being talked about, that for fragmentation at the MS level, because we cannot reduce the peptide levels at the MS levels. And there's this electron transfer dissociations, which transfers the electron ions and the ATD side chain and the position transfer. So this is how some sums you can perform. Again, I think the slides is not moving. I will just do that. So the time of flight mass spectrometer is that. So time is equals to the distance by velocity, tt by v. So have an equation, v equals to 2 root k by m. So now substitute this into t equals to d by v. So d by e root 2 k by m, which is dm by 2k. So the 1 mole of 24, that is mg's molecular weight, or formula, so that has a mass of 254 gram per mole. So to find the mass of an ion, we need to divide by Avogadro's number, 24 by this thing, which is 3.6984 divided, divide this number by 1000 to convert into kg. So 3.98500 kg. Kinetic energy is 2 into 1.0 into 10 to the power minus 6. So 1.2 meters. That the time is required is this much, m by 2 ke, so that is 1.69 into 10 to the power minus 5 seconds. So if you need to calculate the masses and the kinetic energy, mass equals to relative isotopic mass by 6.023. 6 travels to the mass spectrometer with an energy of 1.95 minus 15 joule on seconds to reach the detector. Length of the flight is in meters. So calculate the velocity with the formula and the distance with this formula. So once you get it, we can get the value, right? So try this out if you want to do that. So I think we can have an extra class, and on that extra session we can deal with the genomics and the genomics technologies. I think today we can end at spectrometry with proteomics technologies. And if you have any questions or anything to ask, you can do so now. I guess we can end the meeting if there is no question. So thank you all for joining.